So hello, everybody. It is noon here on the East Coast in Massachusetts. My name is Kate Fitzgerald. I'm one of the co-organizers of the Global IMMUNOTALK Symposium. So I want to welcome everybody here to the first talk of 2023. We have a terrific lineup of speakers for this coming year. I'll introduce today's speaker in a moment, but just to remind everybody, next week we'll have Kenya Honda speaking at noontime Eastern Standard Time on next Wednesday. So it's a real pleasure to introduce today's speaker. So we're joined by James Chen from UT Southwestern in Dallas in Texas. James is an investigator of the Howard Hughes Medical Institute, and he's professor in the Department of Molecular Biology at UT Southwestern, where he's been since 1997. He is also director of Inflammation Research Center, and he's the George L. McGregor Distinguished Chair in Biomedical Sciences at UT Southwestern. So before moving to Dallas in '97, James was a senior scientist at ProScript, where he was involved in developing a protease inhibitor, or a proteasome inhibitor, Velcade, which is now used for the treatment of multiple myeloma. And then James decided to go back to academia. So he joined UT Southwestern as a faculty member and has really risen through the ranks there. His lab has really made seminal discoveries in innate immune signaling in many different areas. I'll just touch on a few. So he's really famous for his work on the role of ubiquitination in protein kinase activation and then NF-kappa B signaling and also MAP kinase signaling. He discovered mitochondrial antiviral signaling, or MAVS, a critical adapter protein that's localized on the mitochondria and important in antiviral sensing and RNA sensing pathways. And then what I think we're going to hear about today is really his world-class discovery of cGAS, this nucleotidal transferase enzyme that recognizes cytosolic DNA and generates a novel second messenger called cGAMP that then propagates inflammation and activates the STING pathway. And James has really done the sort of foundational work in this pathway. He's defined the role of these mechanisms in antiviral defense and also in autoinflammatory and autoimmune diseases. So not surprisingly, James is highly cited. He's incredibly well funded, and he's also been the recipient of many awards. I'll mention a few here. So he's the recipient of the National Academy of Science Award in Molecular Biology in 2012, the American Society of Biochemistry and Molecular Biology Merck Award, the Lurie Prize in Biomedical Sciences from the NIH, from the Foundation of NIH, and then the Breakthrough Prize in the Life Sciences in 2019. He's also an elected member of the National Academy of Sciences and the National Academy of Medicine. So James, it's always a pleasure to see you. We're really excited to hear about your beautiful work for this audience across the globe. But before we get started, and this is really sort of for trainees who like to learn a little bit about some of the famous scientists that they read about in papers, can you talk a little bit about a trait in your personality that's really sort of helped you be so successful? And we'll all take notes so we can become like you.
All right, thank you, Kate. It's very nice to be here. And so for your question, I think that I'm more of a focused person. I like to focus on things, and I get uneasy when I get too distracted, when things get too complicated. So I'm happier if I focus on a single task or one or two tasks. And it's getting more and more difficult because the social media, it's widespread, and these AI technologies are getting better and better and grabbing our attention, more and more addictive, and so it's not easy, but I still like to be more focused. So.
Yeah, it gets more challenging as you get more advanced in your career and probably pulled in lots of different directions. It's impressive to stay focused. All right. Well, let's see if you can share your screen, and then we look forward to hearing about your talk on DNA immunity from human to bacteria.
Okay, thank you.
Wonderful, thank you.
So you can see the slides?
Yeah, everything looks good. Thank you.
All right, okay, great. All right, thank you very much, Kate, for the introduction, and I want to thank all the organizers for the invitation to speak in this lecture series. So today I will talk about some of our recent work on the cGAS-STING pathway. This is the DNA sensing enzyme cGAS, which is activated by binding to double-stranded DNA. And the activated enzyme converts GTP and ATP into the cyclodinucleotide, cyclo-GMP-AMP or cGAMP. This small molecule then functions as second messenger that binds and activates the ER membrane protein STING. STING then triggers a signaling cascade leading to an innate immune response. I want to start with this simple cartoon to illustrate the cGAS-STING pathway. And for those of you who have seen this cartoon, please bear with me. I just want to bring everybody down to my level. So here is a cell that is infected by a virus, in this case a DNA virus. The virus infects the cells, inject DNA into the cells, and this DNA in the cytoplasm is a danger signal that is recognized by cGAS. The cGAS binds to the DNA and becomes activated. This is the crystal structure of cGAS. So when DNA binds to cGAS, it pushes this pink activation loop, and it induces a conformational change in the active site of the enzyme. DNA binding also induces the dimerization of cGAS, which is important for its activation. So now activated cGAS converts GTP and ATP into cyclo-GMP-AMP. This is GMP, this is the AMP part, and it has two phosphodiester bonds. One is 2' 5' link, the other one is 3' 5' link. So we call it 2' 3' cGAMP. And this is a potent second messenger molecule that activates the immune response. So now one molecule of the enzyme makes multiple molecules of the second messenger. Each second messenger binds to a STING dimer on the ER membrane. STING then activates the transcription factors NF-κB and IR3, and these transcription factors move into the nucleus to induce the expression of hundreds of genes that are important in immune and inflammatory response. Here is another view of the pathway. So cGAS is activated by DNA to produce a second messenger cGAM, which binds and activates STING. STING then activates the kinase IKK and TBK1. IKK activates NF-κB, TBK1 activates IR3. These transcription factors function together in the nucleus to induce terminal interferons and other cytokines to fight viral infection. And an important feature of cGAS is that it can be activated by any double-stranded DNA independent of the DNA sequence. And the crystal structure of the cGAS DNA complex shows that cGAS binds to the sugar phosphate backbone of the DNA. It does not have any direct contact with the base pair, and that explains why its DNA sequence independent. And this property allows this single protein sensor to detect a large variety of pathogens as long as these pathogens have DNA. And so these include DNA viruses such as herpes virus, retroviruses such as HIV, bacterial such as MTB, and parasites such as malaria. And another source of DNA that can activate cGAS is our own DNA. Our own DNA is usually in the nucleus and in the mitochondria. But under some pathological conditions, if our own DNA gets into the cytoplasm, it can activate cGAS, and in these cases, it can trigger autoimmune diseases. And one example of the autoimmune disease is Aicardi-Goutieres syndrome, or AGS. And these are the children with the disease. They have calcification in the brain, as shown here. And these are monogenic diseases, and beautiful work from Yannick Crow and others has identified most of the genes that cause this AGS syndrome, and the majority of these gene mutations lead to activation of the cGAS-STING pathway. And for example, TREX1 is a nucleus that usually degrade DNA in the cytoplasm of the cells, and about 25% of these AGS patients have mutations in TREX1 that cripple the activity of this enzyme, and that then leads to accumulation of cytoplasmic DNA to trigger type 1 interferon production. And the question is whether these kids have the disease because they have hyperactivation of the cGAS pathway. So several years ago, we addressed this question in TREX1 deficient mice, and these mice die within a few weeks or months after birth because they have severe inflammation in many different organs. And we asked, if we remove cGAS from TREX1 deficient mice, can we rescue the disease? And so a graduate student, Daxin Gao, generated cGAS TREX1 double knockout mice, and 100% of these mice survived and they didn't have any symptoms of disease. Interestingly, even if we just remove one allele of cGAS, we can largely rescue the survival. And we think that this haploinsufficiency is due to a phenomenon that a student in my lab discovered, Ming-Jen Du, and he is called liquid-liquid phase separation. So Ming-Jen found that cGAS binds to DNA through multivariant interactions, and these multivariant interactions drive liquid-liquid phase separation that leads to the formation of these liquid droplets in which cGAS is activated. And this mechanism renders cGAS activation highly dependent on the concentration of both cytoplasmic DNA and cGAS in the cytoplasm. So when the cytoplasmic concentration of DNA is elevated but still not very high, such as in TREX1 deficient cells, then cGAS phase separation is highly dependent on the concentration of cGAS. And that could probably explain why removing one allele of cGAS, and that is reducing the concentration of cGAS by 50%, can largely rescue the disease. And we think this is quite exciting because it suggests that if one can develop an inhibitor of this enzyme, even 50% inhibition of this enzyme may provide therapeutic benefits for patients with autoimmune diseases such as AGS. Work from many labs throughout the world in the past few years has shown that the cGAS-STING pathway plays a central role in many physiological and pathological processes. This pathway is clearly very important for host defense against microbial infections. As I just showed you, aberrant activation of this pathway can lead to a variety of autoimmune and inflammatory diseases. Activation of cGAS by mitochondrial DNA can lead to neurodegeneration, as shown by Richard Yu and Seth Masters, and these include like Parkinson's disease and ALS. Recently, we showed that activation of the cGAS pathway can lead to a non-canonical form of autophagy, and we and others have shown that activation of this pathway can lead to cellular senescence, and this pathway is also very important for anti-tumor immunity. So that's the introduction. In today's talk, I'm going to talk about two short stories. The first story is on the regulation of the cGAS-STING pathway in animal cells, and the second story is on bacterial cGAS in anti-phage immune defense. So for the first story, we asked the following question. After cGAM is produced by cGAS, how does that activate the downstream signaling cascade? And specifically, we asked, how does cGAM activate STING? How does STING activate TPK1? How does TPK1 activate IL-3? And I'm going to talk about the third question first because of this experiment that we did many years ago. So here, if we infect cells with an RNA virus, BSV, which activates the RIG-I-MAPS pathway, it leads to the activation of TPK1 as well as phosphorylation of IL-3, and that's expected because TPK1 is the kinase that phosphorylates IL-3. If we stimulate cells with herring testis genomic DNA or htDNA, which activates cGAS-STING pathway, we can also see phosphorylation of TPK1 and IL-3 phosphorylation, and that's also not surprising. What is surprising is that when we stimulate cells with interleukin-1 or TNF, now we can see TPK1 activation, but no IL-3 phosphorylation. And that was somewhat surprising because there's overwhelming evidence that TPK1 is the kinase that phosphorylates IL-3. And so the simplest model that could explain this result is that TPK1 is necessary but not sufficient for IL-3 phosphorylation. Now, if we look at these three different pathways that can lead to the activation of TPK1, this is the RNA pathway that activates the RIG-I-MAPS pathway. This is the DNA pathway that activates cGAS-STING. This is the IL-1 pathway that activates this protein called TRIB6 that my lab worked on many years ago. And we showed that TRIB6 is a ubiquitin E3 ligase that can catalyze a form of polyubiquitination called lysine 63 polyubiquitination, which activates IKK through a proteasome-independent mechanism. TRIB6 can also activate TPK1. However, in this case, TPK1 that is activated by TRIB6 cannot phosphorylate IL-3. In contrast, TPK1 that is activated by STING and MAPS can lead to phosphorylation of IL-3. So there must be something unusual about STING and MAPS and their ability to not only cause TPK1 activation, but also cause TPK1 to phosphorylate IL-3. And so we think this puzzle was solved by a former graduate student, cGAS Liu. At the time, cGAS was working on the regulation of MAPS by RNA virus infection, and she found that MAPS is phosphorylated at this serine residue in response to RNA virus infection. cGAS then looked at the surrounding amino acids, and she found this short sequence motif that is highly conserved in MAPS. And this sequence motif is also highly conserved in IL-3, and its phosphorylation of this serine residue, serine 396 of IL-3, that causes it to dimerize and activate transcription. And this sequence motif is also phosphorylated, it's also conserved in STING, and in this, she showed that this serine residue, serine 366 of human STING, is phosphorylated in response to DNA stimulation. And this short sequence motif is also conserved in other proteins involved in innate immunity, such as TRIF. So TRIF is an adapter protein involved in some toll-like receptor signaling, such as TLR3 and TLR4. And what is common among these different pathways is that they can all lead to the induction of type 1 interference. So when each of these receptors is engaged by an upstream ligand, it would lead to the activation of the, sorry, I think my slide is frozen. Okay. Let me, sorry about that, let me start again. Okay, all right. So when each of these receptors is engaged by an upstream ligand, it will lead to the activation of the kinase IKK and TPK1, which phosphorylate this sequence motif. We call the police motif. So now the adapter is phosphorylated. And this phosphorylated motif then recruit IR3 because IR3 contains this positively charged surface. And so now IR3 is recruited and allows the kinase TPK1 to phosphorylate IR3. The phosphorylate IR3 then form a dimer and move into the nucleus to induce type I interferons. So what we are proposing here is that that phosphorylation of these adapter proteins, this sequence motif, provides a licensing mechanism that allows the kinase to phosphorylate its substrate, in this case IR3. And for interleukin-1 and TNF, they don't engage any of these adapter proteins, and that's why they cannot lead to IR3 phosphorylation or type I interferon production. In contrast, with these other pathways that represents virus infection or bacterial infection, they can lead to induction of type I interferons. And also, obviously, you want type I interferon production to be tightly regulated. So we think this provides a sort of unifying mechanism that explains why these different pathways can lead to type I interferon production. But for the experts in this field, you may ask, what about other tolerant receptors that can lead to type I interferon production, such as TOR 7, 8, and 9? And this question was recently addressed by a very nice paper from Julia Sperti-Fergel's lab, and they identified this protein tester, which also had this police motif that we identified. So when these endosomal tolerant receptors are engaged by DNA or RNA in the lumen of the endosome, they will activate a MyADA-dependent signaling cascade, leading to the activation of IKK beta. IKK beta then phosphorylate IRF5, in this case, that leads to the induction of type I interferons and other inflammatory cytokines. However, before IKK beta can phosphorylate IRF5, it must first phosphorylate this police motif of the tester protein, which then recruit IRF5 for phosphorylation by IKK beta. So now back to this cGAS-STING pathway. We think we have a better understanding of how TPK1 phosphorylate IR3, and the bottom line is that in order for TPK1 to phosphorylate IR3, it must first phosphorylate STING. So we just added another question to the list. And to answer all these questions, we feel that we should try to solve the structure of full-length STING. STING is a small membrane protein of about 40 kilodalton. It has 4 transmembrane domains that anchor it to the ER membrane. It has this ligand binding domain that binds to cGAM, and it has this C-terminal tail in which this serine residue, serine 366, is phosphorylated. And the crystal structure of the ligand binding domain of STING has been solved by several groups, but prior to this work, we didn't know any structural information for the rest of the molecule. So several years ago, we assembled a team to try to solve the structure of full-length STING, and these include Chonggang Zhang in my lab, Guijin Sang in Professor Xiewu Zhang's lab, and we're very fortunate to collaborate with Professor Xiaochen Bai, who's a brilliant cryo-EM expert. And together they solved the structure of full-length STING by cryo-EM, which I think is quite remarkable because STING is a small membrane protein. Even the STING dimer is only about 80 kilodalton, which is quite small for cryo-EM. So in any case, they solved the structure of full-length STING, which is shown here. So STING forms this domain swap dimer, each monomer has 4 transmembrane helices. There's this connector helix that connects to the ligand binding domain, and these 2 ligand binding domain forms this cGAS binding pocket. So this is the structure of STING in the absence of the ligand. Now, what happens when the ligand cGAS binds to STING? And so this is what happens. When cGAS binds to STING, it induces a 180-degree rotation of the ligand binding domain relative to the transmembrane domain. It also induces a conformational change such that now we can see this new beta-C structure. And these 2 ligand binding domains come closer by about 20 angstrom. And so now, if we compare the structure of the activated form of STING to its apo form, we notice one interesting difference in this connector helix of STING. So if we zoom in here, so this is the structure in the presence of cGAS. So cGAS binds and induces this 180-degree rotation of STING, and so now this cGAS bound form of STING, that these 2 helices are further apart as compared to the apo structure. In the absence of cGAS, these 2 helices are really close to each other, such that it can accommodate only a small amino acid, such as a glycine. And the prediction here is that if we change this glycine residue to a larger amino acid, it might cause a spontaneous activation of STING. And this is indeed what we found. So here, if we mutate this glycine residue to other amino acids, we can see strong activation of interferon beta even in the absence of cGAS. And similarly, if we look at signaling in the cells, these glycine mutants basically can lead to the activation of the signaling cascade even in the absence of cGAS. In contrast, for the wild-type STING, activation of the signaling cascade depends on the presence of cGAS. And this is very interesting because mutations in these residues have been shown to cause autoinflammatory disease called SAVI, or STING-associated vasculopathy, with onset in early infancy, which was first characterized by Rafaela Gobagninsky. Okay, so we also saw the structure of STING bound to TPK1, which was shown in one of these 2 papers. And based on these studies, we proposed this model. So STING sits on the ER membrane as a dimer, and this is a cGAS molecule that is produced by cGAS. So cGAS binds to STING, it induces a 180-degree rotation of the ligand binding domain relative to the transmembrane domain of STING. And this causes a conformational change such that STING can now form an oligomer. STING then uses the C-terminal tail to recruit the kinase TPK1, and as a result of STING oligomerization, it brings TPK1 close to each other such that it can now autophosphorylate itself, leading to TPK1 kinase activation. And this kinase then phosphorylates the STING C-terminal tail that is not used for the recruitment. So this STING C-terminal tail is phosphorylated. Another STING C-terminal tail is phosphorylated, and this phosphorylated STING C-terminal tail recruit IL-3 and position this serine residue, serine 196, this serine residue 396 of IL-3 for phosphorylation by TPK1. And so this IL-3 is phosphorylated, another IL-3 is recruited and phosphorylated, and the phosphorylated IL-3 forms a dimer and moves into the nucleus to induce type I interferons in other cytokines. So this is a model based on a lot of structural and biochemical studies. And the question is whether this is true in vivo. So a student, Sion Yang, in a lab, used CRISPR to generate knockin mice with mutations at the C-terminal tail of STING. So this serine residue, serine 365 or mouse STING, is the phosphorylation site. And this serine mutation is expected to disrupt the recruitment of IL-3 and prevent interferon production. And however, this mutant can still recruit TPK1 and lead to NF-κB activation, and the autophagy function of this mutant is also normal. Leucine 373 is within the TPK1 binding motif, and mutation of this residue is expected to disrupt not only IL-3 recruitment, but also TPK1 recruitment. And this will lead to the inhibition of NF-κB, but the autophagy induction function is also normal. And we also use other mutants, such as golden ticket mutant, which is like a loss of function of STING. So now Sion have these mice, and she took the bone marrow derived dendritic cells from these mice and stimulated cells with DMXAA, which is a cell permeable STING activator. And so for the wild type cells, DMXAA can lead to the activation of the entire signaling cascade, and it leads to the nuclear translocation of IL-3 and the P65 subunit of NF-κB. It can lead to a strong induction of interferon beta. In contrast, for the S365A mutant, there's no interferon beta production, and that's because for this mutant that the IL-3 nuclear translocation was abrogated, but NF-κB activation was still normal. For the L373A mutant, then there's no IL-3 nuclear translocation, no NF-κB activation. So this mutant is like a STING knockout. And we also look at the RNA-seq data of these cells in response to DMXAA stimulation. For the wild type cells, DMXAA induces hundreds of genes, as shown here. For the S365A mutant, the vast majority of these genes are gone, with the exception of a small cluster of genes here. For the L373A mutant, basically all the genes are gone, very much like STING knockout. And so within these results, validate the model that we propose based on the structural studies. More importantly, this knockin mice provides the in vivo model for us to look at distinct signaling pathway downstream of STING. So we look at three different mouse models to look at antiviral immunity, anti-tumor immunity, as well as autoimmunity. In the interest of time, I'm just going to focus on autoimmunity. And the model that we use is the DNS2 knockout mice. So DNS2 is a major enzyme that digests DNA in the lysosome of our cells. And many years ago, Shige and Nagata in Japan generated DNS2 knockout mice. And these mice are embryonic lethal. The embryos die of anemia, and that's because the embryos produce so much type I interferons they kill all the red blood cells. And the Nagata lab tried to rescue these DNS2 knockout mice by crossing them to toll receptor knockouts, MyD knockouts, etc. Right? They couldn't rescue, but they can partially rescue by crossing them to interferon receptor knockout. And now these mice, the double knockout mice, are born, but they develop severe arthritis, as you can see here. And these phenotypes may be explained this way. So in the DNS2 deficient cells, you have accumulation of DNA in the cytoplasm that can lead to the induction of not only type 1 interferons, but also production of these inflammatory cytokines. And this interferon induction leads to anemia and embryonic lethality, and the induction of these inflammatory cytokines lead to arthritis. And if you take away interferon signaling by knocking out interferon receptor, you can rescue the embryonic lethality, but these mice continue to produce a lot of these inflammatory cytokines that lead to arthritis. Now, if we take away cGAS, and if cGAS is a major sensor of this pathway, we would expect that we will be able to rescue all these phenotypes. And that's indeed what we found. So this DNS2 cGAS double knockout mice, they don't have arthritis anymore, whereas for the DNS2 interferon receptor double knockout mice, they have severe arthritis. Now, what about the STING mutants? So we cross out STING mutants to the DNS2 knockout mice. And here I would draw your attention to these two point mutants. So the S365A mutant of STING will disrupt IR3 recruitment, and so these mice will not be able to produce type 1 interferons, but they can still activate NF-κB. The L373A mutant disrupt TPK1 recruitment, and they will basically knock out the entire STING pathway. So for the S365A mutant, these double knockout mice, they are born, but they develop severe arthritis. But for the L373A mice, basically you can rescue all the phenotypes. So a single point mutation of STING can rescue these lethal inflammatory phenotypes of DNS2 deficiency. And this is seen more clearly with the histology of the bone. The S365A mice develop severe bone erosion, and the L373A mice, they didn't have any bone erosion. We also measure inflammatory cytokines in the blood of these mice, including IL-6 and TNF. The S365A mice produce a lot of these inflammatory cytokines, and the L373A mice, they didn't produce these cytokines. And finally, we asked, can we treat these arthritis disease phenotypes in the S365A mice with a TNF blocking antibody? Indeed, this TNF blocking antibody can basically block the development of arthritis in S365A mice. So to summarize this part of my talk, we saw the structure of full-length STING in its complex with TPK1, which revealed the mechanism of activation of STING by cGAM, as well as the activation of TPK1. And STING mediates immune and autoimmune responses to both type 1 interferon dependent and independent mechanisms. So it's not just type 1 interferon that is important. The type 1 interferon independent mechanisms are also very important for STING signaling. And our results suggest that blocking interferon signaling may not be sufficient to treat the STING-mediated autoimmune diseases. And blocking TPK1 recruitment to STING may be more effective. Now, I'm going to switch gear and turn the clock back by a billion years. And so now I will talk about bacterial cGAS and anti-phage immune defense. Throughout billions of years of evolution, there's constant arms race between bacterial and bacteriophages that give rise to enormous diversity of both. So it is estimated that there are about 10 to the 31st bacteriophages, which is more than
all other living organisms combined, including bacteria. Like animal viruses, phages have both DNA and RNA genomes, with double-stranded DNA viruses comprising more than 95% of all phages. Bacteria have powerful immune defense systems against phage infections. The classical examples include restriction enzymes and CRISPR. So CRISPR is the adaptive immune system in bacteria that fight phage infection. And one of the most exciting development in the cGAS field in recent years is the discovery of a large family of cGAS-like enzymes in bacteria. And work from Philip Kranz's lab identified more than 6,000 cGAS-like proteins in bacteria, which can make all sorts of cyclic dinucleotides and cyclic oligonucleotides. And work from Rotem Sorik's lab has shown that the major function of these cGAS-like enzymes is for anti-phage immune defense. For example, in this very nice paper from Rotem Sorik's lab, they show that bacterial cGAS-like proteins are often in the vicinity of other proteins that are involved in anti-phage immune defense, such as CRISPR. And the bacterial cGAS itself is often organized in operons that contains other proteins that regulate cGAS function. So these cGAS operons are called CBAS, or cyclic oligonucleotide-based anti-phage signaling systems. So when a bacterium is infected by a phage, the bacterial cGAS is activated through an unknown mechanism. And these cGAS-like enzymes convert GTP and ATP into cGAMP. And this cGAMP molecule, in this case, activates a bacterial phospholipase called CAPV. And CAPV then chew up the bacterial membrane to cause the bacterial cells to commit suicide. And that limits the virus spread to protect the bacterial community. And when I read this paper, I was really intrigued by the finding that the bacterial cGAS is very often next to other genes that encode enzymes with ubiquitin E1 and E2 domains, as well as JAP domains, which is a deregulation enzyme. And I was intrigued by this because in my previous life, I was working on the ubiquitin system. So at the time, Justin Jensen joined the lab, and one of the first questions that Justin asked is whether these bacterial E1, E2 proteins, in this case is E1, E2 fusion protein called CAP2, whether this protein can form thioester with ubiquitin like protein in bacteria, much like the mammalian ubiquitin E1, E2. So Justin cloned this cGAS operon from this strain of E. coli to the laboratory strain of E. coli that does not have cGAS. And he epitope tagged this CAP2 protein such that he can do a Western blot to see whether CAP2 can form a covalent thioester with a bacterial protein. And indeed, he found that this CAP2 protein formed a covalent adduct with a 50 kilodalton protein. And this covalent adduct is sensitive to treatment with DTT or hydroxylamine, indicating that it's indeed a thioester. And Justin then cut out this protein and did mass spec, and it turned out to be the bacterial cGAS, which is also called DNCB. Justin then did Western blot to see whether cGAS indeed behaves like ubiquitin and that it can form covalent conjugates. And this Western blot showed that cGAS can indeed form these covalent conjugates in wild type cells. And the formation of these conjugates depend on the E1, E2 activity of CAP2 because mutations of the active site cysteine residues of E1 or E2 abolish the formation of these conjugates. And interestingly, the formation of these conjugates also depend on the C-terminal glycine of cGAS. So mutations of the C-terminal glycine abrogate the conjugate formation. Cap3 is a deubiquination enzyme. A mutation of the active site of this enzyme leads to a slight increase of these conjugates. So cGAS indeed behaves like ubiquitin in that it can be activated by ubiquitin E1, E2 and be conjugated to other proteins in E. coli. And that was quite surprising because cGAS does not look like ubiquitin at all. In particular, the C-terminus of ubiquitin contains two highly conserved glycine residues. For cGAS, like proteins in E. coli, they don't have these conserved glycine residues. Indeed, instead, for those cGAS operons that also have E1, E2 in JAP domain, they have highly conserved glycine and alanine residues. And for those cGAS operons that don't have E1, E2, then there's no conservation. Justin then mutated this glycine of bacterial cGAS to other amino acids, and with the exception of this glycine to alanine, which can still form some conjugates, all other amino acid substitutions abolish this conjugate formation. So now the important question is whether this ubiquitin-like conjugation by cGAS is important for its antiphage immune defense function. Justin look at bacterial lambda-phage infection in this case. So here, this CBAS or cGAS operon can suppress the phage infection very strongly. It reduces the viral titer by more than 10,000-fold. And this activity of CBAS depends on the phospholipase activity of CAP-B. It also depends on the cyclase activity of cGAS. And similarly, mutations of the E1 or E2 activity of CAP2 abrogate the antiphage defense function of CBAS. And importantly, the removal of the C-terminal glycine of cGAS also phenocopy the effect of this E1, E2 mutation. And the mutation of this C-terminal glycine to alanine partially has this partial phenotype because this mutant can still form some conjugates. And the same is true for other phages, including T4 and T6. And so this C-terminal glycine residue is important for the antiphage defense function of cGAS because removal of these residues basically phenocopy the effect of inactivating the E1, E2 activity of CAP2. Now, how does this ubiquitin-like conjugation of cGAS play a role in this antiphage defense function? And so in this experiment, Justin measured cGAM production in cells in E. coli that have wild-type cGAS or have cGAS that is defective in conjugation, as shown in green here. So for the wild-type cells, the phage infection lead to the production, robust production of the cGAM molecule, which is much stronger than those E. coli mutants that cannot form cGAS conjugates. And this suggests that a major function of this cGAS conjugation is to enhance cGAM production. The next question is, what are the cGAS conjugation targets? And we did mass spec analysis and identify many bacterial and phage proteins that can be conjugated by cGAS, but we still don't have evidence showing that that any of these proteins are important targets. Then Justin took an alternative approach, and so he did a forward genetic screen to identify phage antagonists of cGAS conjugation. So for the wild type T4 phage, it can, in fact, E. coli, they don't have CBAS, or have CBAS that cannot have cGAS conjugation. And so Justin mutagenized these phages, and to isolate those phages that are defective in infecting those E. coli with defective cGAS conjugation. And so these phages might have lost the viral antagonist of cGAS conjugation. And so this screen led to the identification of a phage gene called VS4 that encodes a 10 kilodalton protein with unknown function. And interestingly, Justin found that this VS4 protein can bind to cGAM with higher affinity. The KD is about 30 nanomolar. And he went on to solve the crystal structure of VS4 bound to cGAM. And VS4 formed a hexamer that binds to 3 molecules of cGAM. So each cGAM binding site is composed of two VS4 monomers. And so he did this CAP-V activity assay to validate the structural model. So CAP-V activity is stimulated by cGAM, and the wild type VS4, this viral protein, can inhibit CAP-V activation by cGAM. And mutations in the cGAM binding residues basically abrogate the inhibitory effect of VS4, so the mutations that disrupt the organization of VS4 is shown here. So Justin also did bioinformatic analysis to identify VS4 homologs in bacteriophages, and he identified about 200 VS4 homologs in diverse bacteriophages in all of these families. And in particular, in these VS4 proteins, the residues that are involved in cGAM binding are conserved. So based on these results, we proposed the following model. So here is a bacterium that is infected by a phage. And the phage somehow activates cGAS through an unknown mechanism. cGAS then converts GTP and ATP into cGAM molecules. cGAM then binds to CAP-V, activates its phospholipase activity that chew up the bacterial membrane, and that causes the viral infected cells to die and to protect the bacterial community. So in this case, the bacterial won. But for some bacteriophages, they fight back by producing the viral antagonists such as VS4. And the VS4, in this case, functions like a sponge. They soak up cGAM molecules to prevent CAP-V activation. And this allows the phages to propagate. So in this case, the phage won. However, the bacteria want to get an upper hand, and they do this by through this ubiquitin-like conjugation of cGAS that greatly enhances activity to produce cGAM, and they produce so much cGAM they overwhelm the viral antagonist, and the free cGAM molecule bind to CAP-V, and CAP-V then chew up the bacterial membrane to kill the virus infected cells. So this is a sort of a rock, paper, scissors type of arms race between bacteria and phages. And we think that ubiquitin-like conjugation system evolved as a result of this arms race. And I should point out that that this ubiquitin-like conjugation of cGAS was also discovered by Aaron Winnie's lab in University of Colorado, as shown in the archive paper that they posted. So this work, it's really sort of, at the beginning, we still have a lot of unknowns. It basically raises a lot more questions than it has answered. And important questions include, how does cGAS detect phage infection? Unlike human cGAS that can be activated by DNA, there's no evidence that bacterial cGAS can be activated by DNA alone. What are the targets of cGAS conjugation? How does cGAS conjugation activate anti-phage immunity? Are there other targets of ubiquitin-like conjugation enzymes in bacteria? I think our discovery of this cGAS as a ubiquitin-like protein in bacteria is quite fortuitous. So the question is whether there are other ubiquitin-like conjugation proteins. Okay. So finally, I want to go back to this slide just to highlight the fact that cGAS now join restriction enzymes and CRISPR as an important anti-phage immune defense system in bacteria. You can think of CRISPR as a bacterial adaptive immune system, and cGAS as a innate immune defense system in bacteria. And interestingly, we no longer use the CRISPR or restriction enzyme system to protect us against viral infection, but we still use the cGAS pathway to defend us against viral infection. So this pathway has been conserved for over 1 billion years. And finally, I want to thank the people who did the work, the more recent work on dissecting distinct STING signaling pathway downstream of STING in vivo was done by Sion Yang and Tong Li in the lab, and the work on bacterial cGAS was done almost exclusively or single-handedly by Justin Jensen with the help of Tony and Feng Du. And thank you very much. I'll stop here.
Great, James, that was absolutely fabulous. Thank you so much for a terrific talk. Do you mind stop sharing, and we'll just remind people how to ask questions? If Fozia can share her slides, if not, I can do that. All right, let me try. I don't know. She might be. All right. So just to remind our audience members, James, can you see? You can see that?
I can see it. Yes. Yeah.
Okay, good. Yeah. All right. So the way we do this is we do questions and answers through Twitter. So you can search for the Global IMMUNOTALK hashtag, and then find a tweet that says ask questions for James Chen here, and then James will be happy to answer questions that come in through that mechanism. So James, thank you. That was really terrific, really tantalizing, especially the last part. Really absolutely spectacular, beautiful work. So thank you very much. Thanks to our audience, and just a reminder that next week's speaker is Kenya Honda, and we look forward to joining you then.
All right. Thank you very much.